Hello, everyone. Welcome to the OBS seminar today. We have Dr. Zilin Li joining us. He is an assistant professor in the Department of Biostatistics and Health Data Science at Indiana University School of Medicine. Um, Prior to this, before being an assistant professor, he was a research scientist, research associate, and a postdoctoral research fellow in Professor Zihong Lin's lab in the Department of Biostatistics at Harvard T.H. Chan School of Public Health. And he also received his PhD from Tsinghua University in 2016. My name is Raj. I'll be moderating the talk today. So if you have any questions, you can paste them down in the chat. And with that, Dr. Li, the floor is yours to give the presentation.
Yeah, thanks for the introduction, Raj. Good morning, everyone. Thanks for attending my presentations. So my name is Zilin Li, and I'm an assistant professor in the Department of Biostatistics and Health Data Science at Indiana University School of Medicine. So today I'm going to present our project, STAARpipeline, a framework for detecting non-coding rare variant associations for large-scale whole genome sequencing studies. And this work was recently published in the Nature Medicines. So first, I will start with the outline of my presentations. So my presentations includes 4 parts. The first part, I will introduce the background of the large-scale sequencing studies, especially for the rare variant association analysis. And then I will introduce our proposed STAARpipeline and all-in-one rare variant analysis tools for the large-scale sequencing studies. And then in the third part, I will introduce examples for the real data applications. We will apply the STAARpipeline to analyze the Heatmap freeze-5 lipids data. And finally, it is the conclusions of my presentations. So let me start from the background. So for every of us, each human genome contains 3 billion base pairs. Basically, it is the letter ATGC. An important goal of human genetic research is to detect the genetic basis of human diseases or traits. So many of you might have heard about GWAS. GWAS is short of genome-wide association studies, and the GWAS has been widely used to detect the genetic basis of human diseases or traits in the past 15 years. GWAS is an array-based technologies, and one limitations of the GWAS is that it only focuses on the common variants. So here, the common variance is defined as a genetic variance whose malarial frequency is rather than 5% or 1% in the study populations. So let's look at these 2 examples. So assume we have 7 individuals in our studies. Now we look at this variant in the gray circles. We could see that among the 7 individuals, there are 3 T and 4 Cs. So hence this variant is a common variant, and the GWAS data might cover it. So although the GWAS has been successful detecting thousands of common variants associated with human diseases or traits, however, these common variants only explains a small fractions of the heritabilities. And as we know, so this phenomena, this phenomena is called the missing heritabilities in genetics. And recent studies indicate that so this missing heritabilities might be account for the rare variant. And the researchers also found that so the majority of the variants in the human genomes are the rare variant. So the current biobank level data has huge sample sizes. For example, so the UK Biobank GWAS data has about 500,000 of individuals. However, the common variants covered by these large GWAS is still less than 10% in the human genomes, and the remaining 90% of the variants in the human genomes are the rare variants. Recent studies also indicate that so the rare variants have larger effect sizes and are more likely to cause diseases. Researchers also found that the coded proteins of rare variants are more likely to be drug targets. So in order to study the effect of rare variants, a rapidly increasing number of whole genome sequencing studies are being conducted recently. The examples include the transomics of the precision medicine program, which is funded by the National Lung, Heart, and Blood Institute, and also the Genome Sequencing Project, which is funded by the National Human Genome Research Institute, and also the UK Biobank whole genome sequencing data. So by doing the whole genome sequencing, we would be able to sequence every position of the human genomes, which means that so each positions of the 3 billion base pairs. So which makes the data structures of the whole genome sequencing data is very different from the GWAS data. So I will also illustrate the difference use the toy examples. So now we look back to the toy examples. So the whole genome sequencing data will not only include the information of the common variants, which is in the gray circles, but additionally have the information of many newly detected rare variants. In the toy examples, it is just a variant in the orange circles. And we could see that to compare to the common variants, for each of the rare variants in the orange circles, we could find that there is only one individuals have the different alleles compared to the others. So which just indicated that the data structures is very different of the whole genome sequencing data compared to the GWAS data. Then what does the whole genome sequencing data looks like? I will use the TOMAT phrase 8 data as an illustrations. So in total, there are around 140,000 individuals in the TOMAT phrase 8 data, and among these individuals, we found that in total there are around 700 million of the variants. So we could see that the whole genome sequencing data has a huge number of the variants and a very large sample sizes. And we could also find that among these 700 million of the variants, only 1.8% of these variants are the common variants with a malloc frequency larger than 1%, and the remaining 98.2% of the variants are all rare variants. We further find that among these variants, about 61% of these variants are the singletons and or doubletons. For each of these singletons and doubletons, only 1 or 2 of the individuals have the different alleles compared to the others, and these observations just indicate that the genotype matrix of the whole genome sequencing data is very, very sparse. So our question is, now we can see that the whole genome sequencing data is just a large N and a huge P analysis problems, and also related to the sparse matrix problems. So now our question is how could we analyze this huge data set? So the common analysis strategies for GWAS is to perform the individual analysis of each variant, and then do the multiple testing adjustment to detect the significant ones. However, these common strategies could not directly apply to the rare variants because of the lack of power. So in order to solve these problems, the researchers proposed the variant set test. Instead of testing each variant individually, the variant set test evaluates the cumulative effect of multiple variants in a variant set. And when there are multiple variants in a variant set associated with the trait we are interested in, then the variant set test could increase the powers. However, there are still many questions remain to answer when we apply the variant set analysis for the rare variant association analysis in the whole genome sequencing studies. So the first question is just the definitions of the variance set, which is related to the selection of rare variants in the variance set. How could we define the analysis unit to make the analysis powerful for the whole genome sequencing data? And the second question is related to the choice of the variance set test. So in literature, there are many different variance set tests that has been proposed, and we need to choose the ones which are suitable for the whole genome sequencing data. And the third question is about the leverage of the biological informations. So recently, many bioinformatics database has provided the information of the functionalities of the variant. Could we further leverage these information to further increase the power of the analysis in the whole genome sequencing studies? And the last but not the least one is the computation scalabilities. So given the huge number of the analysis, so we needed the computation method as scalable for this huge data set. So my research is mainly focusing on developing statistical methods to perform powerful and scalable rare variant association analysis by addressing these issues. So now we first look at our proposed whole genome sequencing association analysis pipeline, STAARpipeline. So our STAARpipeline is an all-in-one rare variant analysis tools, and the users only need to input the phenotype and the genotype informations, and then our STAARpipeline will automatically handle all the remaining things, including the association analysis and also the results summaries and visualizations. So specifically, so in the first steps, our STAARpipeline will first annotate the variance in the data set. And our STAARpipeline use database favor annotators, use the database favor and the tool favor annotators to functionally annotate any genetics data set and then generate the AGDS file. So here's the AGDS files are a new data format for storing the genotypes and annotations informations. Which and the AGDS files are the all-in-one files that include the information for both the genotype and annotations. So our STAARpipeline also provides some tools to generate the GRMs, which is used to account for the population structures and also the readiness in the following association analysis. Then in the next steps, our STAARpipeline will help the researchers to do the association analysis for the common variants. Just similar as the GWAS, our STAARpipeline provides the single variance analysis. And for the rare variants, our STAARpipeline provides the variance set analysis. So here we considered two different strategies to group the rare variants in our STAARpipeline. So the first one is the gene-centric analysis, where we focus on analyzing the variants in or near genes and groups of variants based on the variant based on the biological functionalities of the variants. And the second one is a non-gene-centric analysis, where we mainly focusing on analyzing the associations in the non-coding genomes, especially in the genetic region. In non-gene-centric analysis, we just group the variants based on its positions. Then after we defined each of the variant set, our STAARpipeline further used the STAAR method to incorporate the multiple function annotations to increase the analysis powers. Then in the final steps, our STAARpipeline provides the tools for some analytical follow-ups. For example, including the summarizations and visualizations of the analysis result, and also STAARpipeline could perform the conditional analysis to detect the novel associations when the users to provide the known variant list that they want to adjust for. So now I will introduce, next I will introduce how our STAR pipeline address the four challenges I mentioned before, and to perform the powerful and scalable real variant association analysis for the sequencing studies. So now we look back to the challenges in the real variant association analysis, and now we looking at the first challenges, which is just the selection of the real variants in the variant set. So our STAR pipeline provide two different strategies to define the analysis unit of the real variants. So the first one is a gene-centric analysis, and the second one is a non-gene-centric analysis. So for gene-centric analysis, we focus on analyzing the variants in or near genes and groups of variants based on as functional categories. So here in the STAR pipeline, we provide 5 different coding categories and 8 different non-coding categories. For the coding categories, we just group the real variants as a loss of function variant, the loss of function variant, and discharge missense variant together, missense variant, discharge missense variant only, and also the synonymous variant. And for the non-coding masks, we just provide the analysis unit of the real variants in the promoters and the enhancers, but overlaid with the cage or the DHS set, and we also provide the mask as the real variants in the UTR regions, upstream regions, downstream regions, and we also additionally provide our analysis mask of the real variants in the ncRNA genes. And then in the non-gene-centric analysis, we mainly focusing on analyzing the associations in the non-coding genomes, especially for the intergenic regions, and we group the real variants based on its physical positions. So our STAR pipeline provide two strategies. The first one is a Zilin window analysis, and the second one is a dynamic window analysis. The Zilin window analysis is the classical analysis approach, which is based on the fixed size moving windows. And for example, we could our STAR pipeline provide the 2 KB Zilin window procedures with 1 KB ski plans. And our STAR pipeline additional provides the dynamic window analysis procedures, which is just use the data-driven size in the analysis and compared to the Zilin window analysis. So the dynamic window analysis procedures could increase the powers by properly select the locations and the sizes of the regions. And also the dynamic window analysis procedures have 2 good properties. The first one, it could dynamically detect the window sizes and the locations. And the second one, it could automatically control the genome-wide type and error rate. So in the following slides, I will introduce the details of the dynamic window analysis procedures. So first, we look at the motivations of these dynamic window analysis procedures. We start from some toy examples. So in these toy examples, we just assume the X labels indicates the positions of each variant, and the blue dot is just each variant, and the Y label is just the effect sizes of these variants. So we assume we have 2 signal regions in the genome, which is just in the red box. When we choose the testing regions are too big, we might lose the powers because we are including some of the noises. So here in the toy examples, we just assume the testing regions and it's a yellow box, and we could see that we might lose power if we use this this testing set because we include 4 more noises compared to the true signal regions. And we might also lose power when the testing regions are too small. Also here in this toy examples, we could see that so compared to the true signal regions, which is the red box. The testing regions, which is the yellow box, might lose power because we exclude some signals so compared to the true signal regions. So now our question is that could we just to perform procedures could dynamically detect the size and the positions of the windows to increase the analysis powers? So to do so, we just to develop the dynamic window analysis procedures using the scan statistics. So here are some mathematical details of the scan statistic procedures. So first, we let the QI be the scan statistics for any region I. And the larger value of the QI indicates the evidence that the regions is related to the trait we are interested in. So here, for example, we could define the QI as the minus log transformations of the p-values for the set-based test. For example, the minus log p-values of the scat or the burden test. Then we define the genome-wide scan statistics Qmax as the maximum values of QI for all the possible searching windows between a range of the searching window lengths. So here is our max, our minimum, the maximum and the minimum searching window lengths respectively. Then we use the genome-wide scan statistics Qmax for testing. So we let H alpha be the 1 minus alpha's quantile of Qmax. And then we could see that the H alphas could automatically control the genome-wide tap-and error rate at the alpha level. So later I will introduce the details how we generate this H alpha empirically. Then if we reject the global null, which means that there is the signal regions in the genome. Then when there are only one signal regions in the genome, obviously the selected signal regions is just the one with the largest region-based scan statistics QI. So when there are multiple signal regions in the genomes. So later I will introduce multiple signal regions detect algorithm to detect each of them. So in this slide, I will just to show you some choices of the scan statistics. So as I mentioned before, so intuitively we could just define the scan statistics as a minus log p-values for the set-based test. So motivated by this idea, so we first to propose the ScanG procedures. So here the ScanGS procedures is just use the scat p-values, and the ScanG procedures just use the p-values of the burden test. And later on, we extend the ScanG procedures to the ScanG star procedures by further incorporating function annotations to increase the analysis powers. So here, so the ScanG star burden procedures is just to use the star burden test, and the ScanG star scat procedures is just using the star scat p-values. And then in this slide, I will introduce you how we generate the empirical threshold H alpha to control the genome-wide tap and error rate. So we just assume the P is just as a covariance matrix of the residuals. So in the first steps, we first generated the pseudoscores U tilde using these formulas. And then in the second step, we just calculated the extreme value Qmax of the scan statistics Qi based on the pseudoscores U tilde calculated in the first step. Then we repeat the steps 1 and 2 for 2000 times, and then use the 1 minus alpha's quantile of the empirical distributions of the Qmax as a data-driven empirical threshold H alpha. And then we could see that as we do that, our H alphas could well control the genome-wide tap and error rate. And also we do extensive simulation studies, and also our real data results all show that this procedures performs very well in controlling the genome-wide tap and error rate. And then when when the test statistics Q max is larger than Qi, we reject the null hypothesis, which means that we have the signal regions in the sequence. Then we will use the following multiple signal region searching algorithm to detect each of the signal regions in the genome. I will also use toy examples to illustrate this algorithm. So in this toy examples, we just assume the X labels is just as the positions, and we assume the black line is the true signal regions in the sequence. So in this toy examples, we have two true signal regions. And in the first steps, we first calculated the Qi for all the regions with the length between the L minimum and L max. Then in the second step, we find all the candidate signal regions with the scan statistics Qi larger than the threshold H alpha, and then select the candidate signal regions with the largest Qi as our estimated signal regions. So in the toy examples, we just have 6 regions with their scan statistics larger than the threshold H alpha, and we call it the candidate signal regions, and which is a blue line in these examples. And then among these 6 candidate signal regions, we first pick the one with the largest scan statistics Qi as our first estimated signal regions. So here it's just this one in the red box. Then in step 3, we first delete all the candidate signal regions overlapped with the true signal regions. Then again, select the candidate signal regions with the largest Qi as our estimated signal regions. So now in this toy examples, as the red line, this red line is our first detected signal regions, and then we remove the two candidate signal regions overlapped with it. And now in our toy examples, there remain 3 candidate signal regions. And again, we find the one in the red box have the largest Qi among these 3, and we choose this one as our second detected signal region. And then in the last step, we repeat step 3 until there is no candidate signal regions. So finally, we just select the regions with the largest Qi in each step as the detected signal regions. So now when we look at the toy examples, our algorithm successfully detects all the 2 true signal regions. So then next we will look at the second challenge in the real variant association analysis, which is related to the leverage of the biological information. So recently there are many bioinformatics database provide the function annotation scores. Where the function annotation scores indicate the biological functionalities of each variant, and then for each different of the function annotations provide the different biological aspects of the variants. So to see them, so first we just look at the correlation heat map for the function annotations. And then from this heat map, we could see that this heat map is just block diagonals, and just for all the annotations in each blocks, which are just indicate the same biological functionalities. So for example, we look at this block. So this block just indicate 8 annotation scores, and each of these 8 scores are all just indicate the protein functions of the different variants. So just to motivate it by this, so we just propose our annotation principle component to do the dimension reductions of the multiple annotations, and also to provide the summaries of of the biological functionalities of each variant. So specifically, so first, based on our prior informations, we just divided the function annotations just into 10 different groups to perform the biological functionalities. For example, the annotations indicates the local diversities, conservations, or the protein functions, and then for each group, we just calculated the first PCs to indicate that biological functionalities. So, for example, for the protein functions, we have 8 annotation scores indicate the protein functions, and then we just calculated the first PCs based on these 8 scores and name it as APC protein functions as annotation as the annotations we want to use to indicate the protein functions. And now we have the function annotations to indicate the different biological functionalities of each variant. And then our STAR pipeline just incorporates these multiple function annotations to empower the variant set test using the STAR framework. So first, we just assume we just use different function annotations as a weight. So here, for example, we use the weight as mallow frequencies, APC local diversities, APC conservations, and also the APC protein functions. And then for each of these weights, we just have a variant set test, and then we just calculated the p-values of the real variant test based on this weight. And then we just use the ACAT method to combine these p-values to get our STAR test statistics t stars, and then we just to calculate the STAR p-value p star just in this simple formulas. And then we have a manuscript to show that. So the calculations for the STAR p-values is fast and accurate. So here, note that our STAR is a general framework and could be applied to any real variant set test. And then the next challenge is just the choice of the variant set, and I will introduce our strategies to pick up the variant set test, which is powerful for the real variant associated analysis in the sequencing studies. So in terms of the variant set test, so these 3 ones are the existing and the most widely used ones. Well, the first one is the burden test. So the burden test just assume all variants are causals with effects in the same directions, and when the majority of the variants in the variant set are signals in practice, burden test performs well. So, for example, when we consider the mask also of loss of function variants of each genes in the gene-centric analysis, so the burden test always works well. However, when there are neutral variants in the variant set, or the effects have the different directions, a burden test will lose power, and the scat works well. So in practice, for example, when we consider the analysis in the non-coding genomes, the burden test always lose powers and the scat works well. And for both the burden test and the scat, assume the signals are dense in the variant set. And when the signals are sparse in the variant set, researchers propose the ACAT-V test, which is powerful when the signals are sparse. However, in practice, we do not know the sparsities of the causal variants in the variant set and also the direction of the variants. So to solve these problems, we introduce our STAR-O test. And the STAR-O test is an omnibus test that combines the burden test, scat, and ACAT-V using multiple annotation ways. So here we could simple to calculate the star O test statistics in this form and calculate our star O p-values in this form. And our star O test has two good properties. So the first is that by incorporating different tests, star O is robust to the sparsities of causal variants and their directions of the effect. And the second is that, by incorporating multiple function annotations, so our star O test is powerful when any of these function annotations can pinpoint out the causal variance. So now we look at the last challenges in the real variant association analysis, which is just the computation scalabilities of the proposed method and the tool. So our STAARpipeline is resource efficient for whole genome real variant association analysis. To achieve this, our STAARpipeline just take the advantage of the sparse structures of the whole genome sequencing data set. So specifically, first, our STAARpipeline uses sparse genotype matrix of real variants to boost the computation speed. And second, our STAARpipeline uses sparse GRM to reduce the computation burdens of fitting the non-mixed models. And also, another big advantage of our STAARpipeline is that our STAARpipeline could automatically define the variant set in the analysis. So finally, in summary, our STAARpipeline just provide a scalable, integrative statistical inference for the whole genome sequencing studies. So when the researchers want to analyze their own whole genome sequencing data set, they only need to provide the data of the genotype and the phenotype data, and the STAARpipeline will just handle all the remained analysis. It will first just to functionally annotate the variance in the data set and generated some user-friendly data format to install both the genotype information and the annotation information. Then our STAARpipeline could automatically define the variant set for the real variant analysis. For the gene-centric analysis, we just group the variants based on its functional categories, and we provide the masks for both coding variants and non-coding variants. And our STAARpipeline also provides the non-gene-centric analysis, which focuses on analyzing the associations in the intergenic regions. And our STAARpipeline provides two different strategies. One is the classical Zilin window analysis strategies, and the second one is our newly proposed dynamic window analysis strategies. And then for each variant set, our STAARpipeline further incorporate the multiple function annotations through the STAR framework to increase the analysis powers. And finally, our STAARpipeline to also provide tools to summarize and visualize the analysis result. And here I also want to say that our STAARpipeline provide a conditional analysis tool, and the conditional analysis could be automatically done in the workflow by providing the known variant list. On the left-hand side, we just show the strategies for the conditional analysis of the single variant, and on the right-hand side, it is the strategies for the variant set analysis. So for the single variant analysis, we need the researchers to provide the known variant list that they want to control for. So for example, the significant findings in the MVP GWAS data set. Then first, our STAARpipeline just do the stepwise selections for the known variant list you choose using our own data set, and to select the independent known variant we want to use in the analysis. Then for each of the detected significant single variants, we just perform the conditional analysis by adjusting on the selected known variants, and then we detected the novel single variants. And for the conditional analysis of the variant set of the real variant sets, so So the framework is the same, but our STAR pipeline to additionally provide options for the users to not only adjust for the known variant list that they provide, but also to adjust for the significant single variant found using your dataset. So the advantages for additionally adjust the list of the significant single variants in your in the individual analysis is that by doing so, we could make sure that the normal findings of the in the rare variant analysis could not be detected by the individual analysis, which means that you detect the novel findings due to the cumulative effect of the multiple rare variants. And then I will show you real data application examples of the STAR pipeline So here we analyze the TOMAT freeze 8 lipids data. So we focusing on analyzing the associations of the total cholesterols. And in total, there are around 30,000 of the individuals in the freeze 5 lipids data. And in total, we are analyzing about 255 million of the variants. So here we focus on the analysis of the rare variant association analysis. So our STAR pipeline provides two different strategies: the gene-centric analysis and the non-gene-centric analysis. For the gene-centric analysis, we provide 5 coding categories and 8 non-coding categories. And for the non-gene-centric analysis, we provide the select window procedures and also the dynamic window analysis procedures. And here is the analysis result for the gene-centric coding analysis. So on the left-hand side, it is the Manhattan plot. So in total, we detect 19 significant coding masks at the genome-wide Buffronic practice level 5 times 10 to the -7, where we just to account for the number of the protein coding genes and also the number of masks. And then from the Manhattan plot, we could see that all of these 19 significant findings are just located in some known lipids associated genes, for example, the PCSK9, ApoB, and also the LDLR and ApoE. And then we do the conditional analysis, and we find that 16 of the 19 associations remain significant in the conditional analysis after adjusting for the known variants from the GWAS catalog at the same genome-wide significant level. So this result just indicate that although all these conditionally significant findings are just located in the known lipids associated genes, but we detected some novel rare variants in these known genes. And also, the left-hand side, it is a QQ plot, which just indicated that our procedures well control the tap and error. And just this slide just show the gene-centric non-coding analysis result. So just similar as a coding analysis result, our STAR pipeline will automatically to provide the Manhattan plot, QQ plot, and also the conditional analysis results. And here, this is the analysis result of the sliding window analysis. Also, similar as the two formal analysis, our STAR pipeline will automatically generate the Manhattan plot of the analysis result, and also the QQ plot of the analysis result, and also the conditional analysis result. And then here for the lipids freeze 5 data, so in the sliding window analysis, in total we detect 59 significant sliding windows, but all of them are also located in some known lipids associated genes, and 2 of these 59 associations remain significant in conditional analysis after we adjusting for the known variants. Here, I want to mention again that these findings means that we detect novel rare variants in or near some known lipid genes. And then this slide just shows the dynamic window analysis results. And then there are two interesting observations The first one is that our dynamic window analysis procedure ScanG star detects 39 significant regions at the genome-wide average level 0.05, and we find that the locations of these findings covers the significant findings of the sliding window analysis. And the second one is that our ScanG star procedures additionally detect one associations in an intergenic regions on the chromosome 9 that is missed by the sliding window procedures. So these two observations just indicate that our dynamic window procedures just increase the analysis powers by detect the locations and the sizes more precisely compared with the fixed size moving windows. And then here the following bullet points just show the details of the novel detected associations that are missed by the sliding window analysis. So here it is the positions, and in total there are around 16 variants in this findings, and the genome-wide error rate is around 0.03, and the related region-based p-values is just around 2.5 times 10 to the minus 9, and then we found there are no known variants in the 1B border regions of these findings, which indicate that this one might be a novel finding. And then here, so this table is just the computation cost of for the STAR pipeline. So assume we have 200 cores, and then we can finish the individual analysis, gene-centric coding analysis, gene-centric non-coding analysis, and the sliding window analysis all within 1 day. Because the dynamic window analysis are considering a much more windows compared to the other analysis, then it requires more of the computation resources. But still assume we have about 800 cores, we can also finish the analysis just in 1 day. So here are some take-home messages for our STAR pipeline. So first, our STAR pipeline provides a general rare variant associated analysis framework for the biobank scale whole genome sequence studies. And the second, our STAR pipeline boosts the rare variant associated analysis power by dynamically incorporating multiple function annotations. And the third, our STAR pipeline performs powerful and resource-efficient computations for biobank scale whole genome sequence studies. And our STAR pipeline is implemented in both the university-based clusters and also the cloud-based platforms. And you could find the softwares just using the two following links. And finally, I want to thanks for the advice for my advisor, Dr. Zilin Li, and all of these works of the STAR and the STAR pipeline are the joint work with Dr. Zilin Li from Zilon's lab. And I also like to thank for the support from the Tom and the Lipids working group, including the Gina from the Boston Universities, Pradeep from the Broad Institute, and the Jerry from the UCLA, and also the Kirsten from Michigan. And thanks for your attention. And any questions for my presentation?
Thank you so much, Dr. Li, for your talk. And like Dr. Li just mentioned, if anyone has any questions, I see that Kuan has raised his hand.
Sure. So I'll take the privilege of asking the first questions when people think. Certainly, that's a wonderful talk and a lot of work you do to optimize OANs of rare variant tests. I wonder, and you may have touched on this. So your STAR pipeline, in a way, have multiple annotation methods that you combine, have multiple tests that you combine with the STAR test, and then I guess in the sliding window, you decide on the best window. In the choices of all of these methods, does it always work best to determine, does it ever work best to determine one optimal way, or your way of combining all these different annotations or almost always work the best?
Because in practice we do not know which annotations could help in analysis, so we recommend just to incorporate the different annotations that represent for the different biological functionalities, and if any of them could help in the analysis, and then our STAAR test could increase the analysis powers. So in summary, in our STAAR pipeline just recommend you to incorporate all of these different annotation pieces. But if the researchers have the have their prior knowledge about which annotations they think works well for their specific analysis, and then they could just to incorporate these specific ones.
I see. Yeah, because that's getting to what I want to ask next is that in a way there could be endless annotations, right? And will that be better or worse when when we keep accumulating different layers, especially on the epigenetic side for non-coding variants?
I think it just depends on the prior knowledge of the researchers. If they just think they have the prior knowledge that these annotations could help and might not help, and then they could include or exclude by the users. But in our side, we think in general we do not know which one might help. So in the pipeline, we recommend you to include, but the STAAR pipeline have the flexibilities to select any of the annotations you want to use or you do not want to use. Yeah.
Yeah. Okay. Before I see other hands raised, then is that the most recommended way you suggest to incorporate like multi-omic information, like epigenome, transcriptome information, or do you explore incorporating them using other methods?
Yeah, so in general, in our STAAR pipeline, when focusing on the non-coding regions, we just to recommend you to incorporate for the different aspects for the epigenetic scores from the multi-omics data, and also. And then also Eric also in Zil's lab, he is just focusing on to extend our STAAR and the STAAR pipeline to the cell STAAR framework, which allows you to incorporate the cell type specific and the tissue specific annotations to further increase the analysis. But they are just in progress.
I see. That's great. And then I guess that's connecting to that. Is there thinking of connecting it and reversing the other way to use it to predict, say, gene expression QTL analysis?
That's a very good question. So this is our future work. We want to further to incorporate to extend our STAAR framework to the QTL analysis.
Yeah, I think that would be so helpful for the field. Thank you so much.
Yeah, thank you.
I have a question, little off topic. So for doing disease risk prediction or any phenotype prediction, one may suggest using both common variants and rare variants. So in such scenario, which strategy of annotation method is best for annotating rare variants with diseases so that they can be combined with the common variants for prediction analysis?
Okay, so if I understand correctly, so your question is about just some kind of related to both incorporate the informations of the rare variants and the annotations in the calculations of the some kind for the polygenic risk scores, for example, right?
Right.
Yeah, I think that's a very good question, because to my knowledge, currently, I do not see any of the procedures. They are just, for example, for the polygenetic risk scores, they are proposed to incorporate the information of the rare variant. But I think this is a very interesting future work to borrow the idea for the STAR pipeline for the association analysis, and then to the polygenic risk scores. But I do think that if you incorporate the information of the rare variants properly, that you could increase the powers, because I think recently Dr. Peter Wischer's group just have a Nature Genetics papers that indicates that if you incorporate the information of the rare variant, you could just to increase the heritability estimations. So we think if you could incorporate the information of the rare variant, that you could improve your polygenic risk score calculations. But by now, I do not see any work there. So I think it is a very good future direction.
Are there any other questions? If not, I wanted to ask a question myself. So within the pipeline, you mentioned that the pipeline uses genotype and phenotype information and then uses its own annotator, the favorite annotator, for the functional annotation of those genotypes. Is it possible for the pipeline to incorporate other annotators as well, like ANNOVAR, for example?
I think our favorite database has already including the including the ANNOVAR informations, and then for the other annotation database. So if many of the users just think that it is very important, and then we will continue maintain the database of the favorite database. So if many users think that annotations it is important, and then we will update a new versions of the favorite database that include that annotations. But if it is only some specific requests from some users, and then they can just to use the framework of the favorite annotators just to add their annotations to the AGDS file. But in summary, which means that first we will continue to update to include more annotations, and the second is that so our favorite annotator is also very easy to extend to incorporate more annotations. But if in that ways it requires some works from the users.
Sounds great. Thank you so much.
Hello, I have a question. For the gene-centric analysis, how you select the different window size? You can choose any window size, but which one is better?
Oh, it is because we are just to detect the window sizes just based on the p-value. Because here you can see that this is a detailed choice of the scale statistics. So basically, we are just because we are consider the different positions and the different size of the windows, and then we just choose the ones with the smallest p-value.
So each window size has a p-value. So you choose the smallest, the window size with the smallest p-value.
Yeah. And not just it is just a size and the positions. And for example, so if we have two of the findings in the dynamic window analysis procedures, and then the size of these two findings also might differ, because maybe for the first findings, for example, maybe for the first findings, 1 KB is the best size, and for the second findings, the 3 KB is the first size. And by our dynamic window analysis procedures, we could use the data adaptive sets for each of the regions, which means that for each of the significant findings, it might have the different size, and the sets could be varied.
Okay, thank you.
Mhm.
I think if no one has other questions, I think we're approaching the end of the seminar. Thank you so much, Dr. Li, for your time and hosting a great talk. And for other open box science seminars, you can see the social media links in the chat. And with that, everyone have a great day.